wanted to share with you today a little bit about our work on the catalysis and the modern applications of catalytic reactions, the discovery of the new catalytic reactions, and their use in biology and medicine, in drug discovery, and also in materials science. And some of the chemistries and the main tasks of modern chemistry, modern organic synthesis, is to find new reactions, new tools to manipulate different starting materials which are available to us, and to study, of course, complex biological systems, study complex materials, to study in general complex systems by means of chemical tools. And catalytic reactions in that sense offer a lot of potential for studying and for interrogating biological systems in particular. Of course, all of the catalysis in nature is part of life. It's all of the transformations which are happening in the context of a living organism are usually catalyzed by enzymes, by very large catalysts which are proteins. So in the lab, of course, we are starting from very different materials, and we are starting from very different tools as well. And that's the art of the modern organic chemistry, and being an organic chemist, to be able to apply those tools, to use those tools for studying the fundamental chemical reactivity, for looking at the modifications of the, for example, as in this case, modification of the natural particles, viruses, building very complex but very well-controlled, well-behaved structures such as dendrimers or dendronized polymers. Those materials can be used for targeted drug delivery, for targeted imaging, for cellular studies, also for targeting, and also for labeling and for studying the actual molecules of life, nucleic acids, proteins, carbohydrates themselves, and then looking at the real-life applications for targeted delivery of those materials, whether it is a drug or an imaging agent, to the tissues, to the specific tissues, like in this case, into the tumor in a mice model. Also, for example, visualizing biochemical processes as they are happening in real life and real time in the cells without fixing them, i.e., in other words, without killing them. How can we become the bystanders? How can we look at those processes that are happening in real life and not interfere with them? Because as you will see, all the tools that we've learned to use and developed over the last 50 years or so, or even probably 100 years or so, are not necessarily very useful in the laboratory setting. And now materials is a whole special area looking at the polymers, functional smart materials polymers, which can work in the structural applications and biomedical applications as tissue replacements, as joint replacements, as actual matrices for growing biological tissues. It's worth looking at the starting materials which nature and which chemists people use in their daily life, and where they're coming from and what tools people are using to manipulate them. And those tools and the sets of starting materials are drastically different. And therefore, we've learned to manipulate those specific ones which we obtain from natural sources, such as our most abundant source is oil, natural gas, hydrocarbons, shown in black here as black chains of carbon-carbon bonds, not really derivatized with any heteroatoms, usually containing just carbon-hydrogen bonds. When those are converted by petrochemical industry, by cracking into olefins, they are endowed at that point with about 24, 26 kilocalories of energy. And then we can climb down the ladder and obtain many more different products which are used in everyday life, starting from polymers to materials to detergents, fertilizers, and even some of the first pharmaceutical compounds, some of the first drugs which were discovered from the natural sources, from coal and gas, not from living things. And the very first patent, incidentally, for cracking was issued to Vladimir Shukhov, who was working in Moscow, although the first refineries were built by Rockefeller family in New Jersey in 1910-20s, about 100 years ago. And during those 100 years, we've learned not only to obtain fuel for cars and other, to refine raw hydrocarbons as energy sources, we also have learned to convert them to all the substances and materials that surround us. In fact, about 90%, over 90% by weight of all chemicals that are produced on a planetary scale are obtained from petrochemical industry. Nature and pharmaceutical industry took a slightly different pathway, although in the beginning they were quite similar to each other. Salicylic acid was one example of the very early drug, was the first antipyretic, the compound which was used by Hippocrates over 400, at about 400 BC to reduce fever and to reduce pain. Then many more years later, in 1897, German company Bayer discovered that acetyl salicylic acid, the introduction of the small acetyl group into the salicylic acid, resulted in a new drug called aspirin, with which you are all familiar, of course, very well. And it has been a venerable drug for the last 100 years for a variety of different diseases and conditions. In this case, the material was already obtained not from a natural living source, not from a plant. It was actually acetylated and by a process of acetylation of a molecule which was obtained from coal industry, but again from hydrocarbon industry. The next example and the biggest revolution in the pharmaceutical world took place in the 1920s, 1930s. When we look at the antibiotics, the very first antibiotics were sulfur drugs, which were discovered incidentally. It was a byproduct of the dye industry, of the colorant industry. People started to use colors and dyes for coloring clothing, for example. And that or they were called aniline dyes, and still are called aniline dyes, the compound shown here on the left with the two nitrogens connected by a double bond. And the first sulfur drug, the antibiotic Prontazil, was called red streptocide, which some may remember, was discovered again by Bayer chemist in 1932. Completely synthetic molecule, not made from any living creature or living source. The white streptocide was later determined to be the sulfanilamide, was discovered and determined to be the active substance. And in fact, some of those sulfur drugs are still used. In parallel, another drama was developing in England, and it was a very interesting time for chemistry and biology and the whole future of the humanity, actually, because Fleming discovered penicillin at that time. 1928, when the first culture and isolation of penicillin was reported, or when he did it first. It took 14 years until 1942 until it was introduced into the clinic. It was not incidentally the first antibiotic. Sulfur drugs were the first ones and were able to cure infections which were considered to be uncurable until that time. Over the last 70 years since the discovery of penicillin, chemistry took a very drastically different course. At that point, we started looking at the natural substances and we wanted to learn how we can make them more efficiently, how we can make it better than nature does. And we developed a multitude of different methods to prepare those compounds with very complex stereochemistry, like one of the latest examples, meropenem, a very broad spectrum antibiotic from AstraZeneca, which was introduced on the market in 1996. It contains 1, 2, 3, 4, 5, 6 chiral centers, contains very unusual bond connections, and it takes quite a bit of time to prepare it. The reason for that is not that unusual and not really unexpected. Like I said, like I said, chemists are starting from a very different set of starting materials. Those are hydrocarbons. Nature, on the other hand, manipulates with carbon dioxide as a source of her carbon blocks, ammonia as a source of nitrogen, phosphate as a source of phosphorus, and oxygen obviously is a product which is obtained by photosynthesis. But more importantly, for natural purposes, it's not oxygen, it's carbon-carbon frameworks, it's carbohydrates which are obtained by photosynthetic processes where energy of the sun is used for those purposes. In reality, if we try to mimic the very same process, the same synthesis in the lab, we will not be able to do that very efficiently, because if we mimic nature and actually try to use the very same starting materials as she does, we'll have to use completely different reagents because we don't have those enzymes, we don't have those big biological molecules which allow us to convert carbon dioxide, water, and everything else what's available to us into the molecules of interest, into complex natural products. A solution to that could be something which we will discuss in the further lectures or we'll look at in the next vignette.